So I'm Kiva, and I'll be speaking about the Australian Reference Genome Atlas, or ARGA. And essentially what it is an indexing platform. So it aggregates data from multiple places online and brings it back to the one place. And the data that we're focusing on is for Australian species and species relevant to Australia, and it's for eukaryotic organisms, excluding humans and excluding model organisms. And just quickly, I'll give you a bit of an overview very quickly, and then we've got a bit of like a video demonstration. So a bit about who we are. We're a joint project between the Atlas of Living Australia at CSIRO, the Australian Biocommons and Bioplatforms Australia. We're an NCRIS-enabled facility, and we have some funding from the ARDC. So why build ARGA? So we heard from life sciences researchers in Australia that they were finding the discovery process for genomics data kind of difficult and frustrating. And part of this was because they had to access multiple different repositories to try and find the data that they were interested in. And as we know, genomics data is important for a really broad range of applications, including primary industries, agriculture, conservation and emergency planning and management responses. So what we wanted to do was improve that genomic discovery and access process for Australian researchers in Australia. And part of what we wanted to do was integrate this genomic data back into the biological context from which they were derived, because often when genomic data is deposited online, it loses a lot of that kind of taxonomic, ecological, and phenotypic information, which is really relevant and can help researchers to discover and understand more about the sequence data itself. And just quickly, our project trajectory. So we started about halfway through last year, and up until about halfway through this year, we were in that design and build process. And then we did some pre-launch testing in July. We had a kind of an early MVP release at the beginning of November, and we're planning a full release by the end of this month, when we're planning to make the URL accessible so that it's available for you to use. But firstly, kind of what we did at the start was we had a consultation phase. So we spoke with life science researchers in Australia about their processes when they were looking for data and accessing data and what kind of challenges were involved. So we spoke to kind of a range of people in a range of different kind of research areas. And from that kind of consultation phase, we derived these kind of three community driven aims. And they're all around kind of the trustability of the data. So we found that when we spoke to researchers, there was this kind of common issue that when they could find data, there was kind of issues around whether or not they could trust it. And that kind of boiled down to three main features. And that was firstly, the taxonomic certainty. So whether a sequence actually was from the taxon it says it was. Secondly, data quality. So the actual quality of the sequence datum. And thirdly, metadata sufficiency. And that kind of relates to the above 2 points. If there's not enough metadata relating to how a specimen was collected and identified, it's hard to have taxonomic certainty. And if there's not enough information about how a sequence was derived in terms of like what platform was used, what technology, what algorithms, it's hard to make judgment calls about the quality of that sequence. So we kind of kept that in mind when we were building ARGA. And we wanted to resolve that taxonomic uncertainty, so we used the National Species List and the Australian Faunal Directory as primary name sources. We also wanted to include informal taxa. In terms of quality assessment, we built some visualization tools to look at kind of raw data and get an idea of, I guess, the quality of that data. And we wanted to address metadata insufficiency by building links with ALA records and specimen collection records, as often those have more information about the kind of collection and identification of a species or a specimen from which a sequence might be derived. So something else we did was we kind of plotted out the data that we were looking at, the genomic data, and really the takeaway here is just that there's a lot of data types and they're really interconnected. So you've got lots of genomic data, you've got related transcriptomic, proteomic, trait data, and all that data is derived from a specimen, and then that specimen has a relation to the species concept. And so we kind of wanted to keep that in mind when we were building the data model for ARGA. We wanted to respect that hierarchical nature. We also wanted to integrate multiple source genomic repository structures. So different repositories are structured differently and they have different terminology. They have metadata terms which might be similar but name something else. So that's something we had to keep in mind. We also wanted our data to be interoperable with biodiversity data, and we wanted to be able to standardize and unify that data under a Darwin Core compatible schema, with Darwin Core being kind of the main ontology that's often used to describe biodiversity data. And the kind of model that we arrived at was this event model. And the way it works is it kind of tracks every event that could happen between the collection of a specimen to the deposition and reuse of a sequence datum. And for each of those events, there's a separate set of metadata which is going to be relevant. And we structured it this way so that we could try and accommodate for as much metadata as possible. We didn't want there to be any data loss when we were bringing in data from multiple sources. One of the key issues that we did have when doing this was because there were multiple sources that we were accessing and they were using different vocabularies and different ontologies, we kind of had to build a model which was able to integrate all of those. So whether it's biodiversity data in the Darwin Core format, or whether it's genomic data and it's using MIXIS, so minimum information about any sequence terms, or some other kind of terminology, we had to build a schema that was able to integrate all of that information. So what we're doing at the moment is we are continuing to integrate different repositories, mapping all of their kind of internal schemas to our own. At the moment, we've got BPA, so that's Bioplatforms Australia's data portal, that data, NCBI, GenBank data, and BOLD within ARGA. And we are also bringing in that biodiversity data in Darwin Core format. And just quickly, a demonstration. So this is a video. That's the homepage of ARGA. So you can go to the search bar and you can type in clover, for instance. So it does pick up on common names. You get these kinds of species report cards for the 4 species that come up if you search for clover. You get that taxonomic breakdown and you can see what data is available, genome assemblies and genetic loci. It takes you to the genome assembly page if you click on Trifolium, and then you can browse the genomes, and then you can click on a specific genome. So that's this one here. You can kind of see more details about it. And then going back to the kind of species page, you can browse along the top. There's different kind of data that's relevant to that species. So clicking on the taxonomy tab, you get that kind of taxonomic breakdown again. And all of those different taxa are clickable. You get the kind of summary page for each taxa if you click on it. So this is clover, or the Trifolium genus. And it tells you what species there are genome assemblies for, how many species are in that genus. So that's 9% of those species have genome assemblies, and 71% have kind of any genetic data. And it gives you a breakdown of which species have the most of that data. And then if you click on that one, so that one was the species with the most genome assemblies. And you go to the genome assemblies, so that is a breakdown of the genome assemblies. The first one up there is the RefSeq genome. And you can click on that and see more information. You can save it to a list. And then if you click on that list, you can see what kind of sequences you've saved. And if you click on that, you get directed to the FTP server for NCBI, so long as it's an NCBI sequence that you've clicked on. And then back to the Trifolium genus page. And you can browse and see what other species are in this genus. And you can just see quickly whether there's a genome assembly available or any genetic loci data available for those species. So clicking on another one, you can look at that genome assembly again, see more details. Again, you can add it to your list and it gets saved to that same list. And then you can continue browsing. And we've run out of time, but this is essentially the, I guess, the core of what it does. There's some other things like functionality and trait filters. And there's also the specimen tab, which will give you a breakdown of what information is available for the different events in that for that specimen. So I'll stop there because I can see I'm being looked at. Yeah, and those are just some of the future directions. We're planning to ingest more data and bring in more kind of traits, filters and that kind of thing, and more community engagement going forwards in terms of generating kind of more standardized terminologies and ontologies. Thanks.
That was amazing. Thank you so much for that. Does anyone have any pressing questions? I've got one just quickly. So in terms of the uncertainty around the taxonomic information, did you find a large percentage that was the case?
Do you mean like of the data that we're accessing?
Of the data, yep.
It's hard to say, I guess, how many are definitively like incorrectly identified. It's more just something that we heard when we spoke to researchers, is that they'd kind of had these kind of instances where they'd used data, they've accessed data and they've analyzed it, and then they've realized, oh, that does not belong to the species that it says it is.